Bekari, and today I'm going to be presenting on Polly and colleagues' paper on how cerebellar RTMS and PAS effectively induces cerebellar plasticity and potential cerebellar plasticity induced by non-invasive brain stimulation. Quick review, cerebellum is an important relay in motor networks and has a crucial role in movement execution and control. It facilitates movement by detecting and correcting errors that occur in the course of the movement, thus making the movement more fluid. These corrections are made via not only the cerebello-thalamocortical pathways, but also via the basal ganglia and brainstem connections. However, in this study, we're only going to be focusing on those cerebellar pathways. Basically, non-invasive brain stimulations have been known to induce plasticity in different areas of the brain, and Polly and colleagues wanted to see if there was a similar effect present in cerebellar tracts. So what they're doing is they're investigating the effects of 4 different types of non-invasive brain stimulations in the same group of healthy participants, and measuring cerebellar plasticity as well as M1 excitability in pathways such as the ones that connect the dorsal motor to motor, cerebellum to motor cortex, those interactions. And they're doing this with the aim of determining the effectiveness of these measures and their potential usefulness in clinical settings. So the main aim of this study is to do a direct comparison of the different types of NIBS techniques in the same group of healthy participants to see if there's an effect on cerebellar plasticity and M1 excitability. There's actually 2 experiments that are examined in this study, and the reason for that is because the first experiment looks at the order of the 4 plasticity techniques, and those techniques were randomized and they were applied at least 1 week apart from each other, pre and post plasticity induction, cortical excitability, um, resting motor threshold, active motor threshold, motor evoked potential. Those were all measured pre and post NIBS, so pre and post stimulation. And what they noticed in the first experiment was that PAS reduced cortical excitability determined by the motor evoked potentials, whereas RTMS increased motor thresholds and facilitated dorsal premotor, motor, and cerebellum motor cortex interactions, whereas TDCS and CTBS had no significant effects. And since they didn't have any significant effects, what they did is they removed them from the second experiment. And then in the second experiment, they similarly wanted to compare they wanted to reevaluate the efficacy of RTMS and PAS, complemented by a sham stimulation as a control condition. And similarly, they compared the pre and post measurements of resting motor threshold, active motor threshold, and motor evoked potential after each technique. In the second experiment, what they found was that MEP amplitudes increased after RTMS and motor thresholds after PAS. An analysis of all the participants who received RTMS and PAS showed that MEP amplitudes were reduced after PAS and increased after RTMS. And also, RTMS caused facilitations of the dorsal premotor, motor cortex, and cerebellum motor cortex interactions. So there were 3 main findings of this study, and the first main finding was that RTMS increases excitability as reflected by MEP amplitudes, whereas PAS decreases MEPs, and CTBS and TDCS were not effective in inducing cerebellar plasticity. The second finding is that RTMS facilitates the interactions between the dorsal premotor cortex, as well as the primary motor cortex and the cerebellum, all of those interactions. And then the third finding is that our results were actually, and this is something that they wanted to improve in further experiments or in future experiments, is that the results from Polly's experiment, they weren't very robust, and they noticed that they would become more clear after the sample size increased to 38. So whereas results were quite variable and equivocal with only 28 participants. So these findings were have considerable consequences for future studies, specifically if there are patients that face lesions in any kind of cerebellar pathways. Um, but anyways, the take-home message for this is that cerebellar RTMS and PAS can effectively induce plasticity in cerebello premotor motor pathways, provided that samples are large enough to be studied. Anyway, I hope that you learned something today, and I thank you so much for listening. Have a wonderful day.